Good morning, everybody. Are you able to hear me?
Yes.
Okay, I expect nursing, physiotherapy, and dental. Are they all on board? I can see physiotherapy, Awinja, and the nursing, Julia. I'm hoping everybody was informed of the time and the introduction of the class. So we'll go ahead. If you are not able to hear me or I'm very fast, please, by show of hands, let me know. Class rep can let me know, okay? And then I would request that everybody mute their microphones. Fine, we are going to deal with the respiratory system. And for this particular course, which is a combination of both hematopoietic, that's blood, lymphoid organs, and also respiratory system and cardiovascular system. So we are going to deal with all of them. And most of our classes, because of the large numbers, will be online. We will, however, have some practicals in which we will arrange you in possibly 3 or 4 groups, and we'll be conducting practicals both in blood and lymphoid organs, the cardiovascular system, and also respiratory system. Now, you will note that all these systems are very closely allied together. You need blood to be able to have the cardiovascular system functioning. The cardiovascular system will pump blood to the body, and in the process, it's also pumping blood to the respiratory system for the purpose of oxygenation. And then the blood vessels will be conducting that blood to all the parts of the body. Now, for the blood, we have both the fluid, plasma, and the formed elements of blood. We are going to look at these formed elements of blood, what they are, and also what is their structure, the structure of the blood cells in terms of their function. So we talk about structure and mention function, although physiology will be dealing with the functions of these particular cells. For the cardiovascular system, we will take our journey from the heart. And remember, we are building on what we have done already in cell biology. When you did cell biology, you looked at the tissues, you looked at muscle tissue, specifically skeletal muscle, a striated muscle, in which you divided it into skeletal muscle and also cardiac muscle. So you know the structure of a cardiac muscle. That muscle, which is a tissue, then forms the organ that we refer to now as the heart. So we'll be looking at that heart, its organization, functional structure of the heart, specifically the heart wall, and also the various parts of the heart in terms of the chorda tendini. We'll be looking at those with the valves. We'll be looking at those in histology. And then we look at the blood vessels in terms of structure. right from the vessels leaving the heart and the ones subsequently returning blood to the heart. So we'll be looking at all those together in a nutshell. And then after that, we look at the respiratory system from the nasal cavity all the way to the lungs for the purpose of oxygenation of blood. So we are going to look at all these aspects in terms of the, when physiology talk about compliance of the lung, what does it mean functionally, histologically? So what we are going to look at is the structural organization of the respiratory system, structural organization of the cardiovascular system. That is what we look at. And in each of these, as we look at them, we'll also be looking at the embryology, the development of the various organs, development of the lungs, development of the heart. We'll be looking at all this. But before we look at the development, I prefer that we look at the structure, the histological structure, and then go back to look at the development, how this has developed. Some aspects of anatomy will be covered by some other people in gross anatomy. So you will also be having practical classes both in gross anatomy and histology. And in the gross anatomy, you'll be looking at the heart, looking at the blood vessels in terms of their distribution in the body, and also looking at the thoracic cavity, where you have the lung and the heart, in terms of the organization of the thoracic cavity to facilitate the respiratory function. So those are the aspects you'll be looking at during this particular session. Various sessions that we're going to have in histology. If you have any queries, please, you can always put it in the footnote so that we can be able to respond. I'll be addressing the class rep, will be the liaison, to let me know if you are getting it clearly or not. Okay, class rep, is it clear? Class rep of dental, are you getting me?
Yes, it's clear.
Okay. So we'll be starting, I'll go back to embryology, but I would like us to start with the histology of the respiratory system. We'll go back to embryology, but I'd like us to start with the histology, and then we'll go back to the embryology system. We will have known the various parts of the respiratory system so that we can now see how these parts have come to develop. The respiratory system, we usually divide it into, I prefer to divide it into three parts. We have what we call the conducting system of the respiratory system. The conducting system, its main function is to conduct air, to allow air to pass to the lungs. So it should not be blocked, it should not collapse. So it should be a rigid structure that will allow air to pass to the lungs and yet might also regulate the flow of that air. During that conduction, there'll be also conditioning of air. We'll be looking at what conditioning is and what structures are adapted for that particular purpose of conditioning. So that is the conducting portion of the respiratory system. Once the air gets into the lung, the deeper parts of the lung, which we'll be talking about, what we specifically call the respiratory acini, or the functional unit of the lung, then we refer to that portion as the respiratory portion. It's called respiratory portion because in that particular area, air is supposed to pass into blood, diffuse to the blood, and carbon dioxide diffuses out of the blood to be removed in the conducting system. So those are the two key parts in terms of the respiratory portion, the respiratory system. We also have what I refer to as the ventilatory portion, ventilatory division of the respiratory system. And the ventilatory division of the respiratory system merely facilitates the intake and removal of air. Intake and removal of air. And those are the structures, chiefly the diaphragm plays a very important role as the ventilatory portion. We also have the rib cage, in which you have the intercostal muscles playing a very major role also in the ventilatory part. Apart from those, we also have accessory muscles that attach to the rib cage or the sternum that influence the diameters of the thoracic cavity. And therefore, we refer to those also as components of the ventilatory portion of the respiratory system, or ventilatory division of the respiratory system. So I've said you have three parts or three divisions. We have the conducting division, merely facilitating air intake into the lungs. We have the respiratory division, where you have the actual exchange of gases, exchange of gases. Then you have the ventilatory part, which facilitates that the air getting into the lung and the air getting out of the lung. So those are the most important parts that we'll be looking at. But remember that most of the systems are integrated together. They're interdependent. So the respiratory part is dependent on the circulatory part. So the circulatory part provides blood to the respiratory system, and that we refer to as the perfusion of the lung. Perfusion. Unlike what the lung does, which is ventilation. So there's ventilation, the air gets in, and there's perfusion, the blood getting into the lung. So that has to be matched so that we have a proper match of blood and air, so that you can have the actual exchange of gases in the respiratory portion of the respiratory system. So we are going to look at the functional structure, functional organizations of these three divisions of the respiratory system. For the conducting part, these will include the nasal cavity, which is normally divided into two parts. The outer part, which is continuation of the epidermis and the dermis, which we refer to as the vestibule of the nasal cavity, and also the nasal cavity proper. The nasal cavity proper. So there's a transition between the vestibule and the nasal cavity proper, in which the epithelia changes. The epithelia changes. Also, the underlying connective tissue changes. Then after the nasal cavity, we have the pharynx, usually divided into three parts. We have the nasopharynx, continuations from the nasal cavity. We have the oropharynx, continuation from the oral cavity. And we have the laryngeal pharynx, which now is a continuation from the larynx. So those three parts constitute what we call the pharynx, and we'll be talking about the structure of the pharynx in terms of the histology. Then we have the larynx, and the larynx, of course, you are aware, consists of the voice box or the codes, voice codes that produce sound. So apart from producing sound, it's also important in conducting air. Then you have the tracheobronchial tree. The tracheobronchial tree will continue from the trachea, the primary bronchi, the secondary bronchi, the lobar bronchi, segmental bronchi, the bronchioles, up to the terminal bronchiole. Up to the terminal bronchiole. That is what we refer to as the conducting system or conducting division of the respiratory system, up to the terminal bronchiole. So its main purpose is merely to facilitate air passageway into the respiratory portion, or respiratory division. Occasionally, you might have blockage or narrowing of these pathways, in which case, if there's a narrowing or a blockage, then you will have less ventilation to the lungs, less ventilation. And like we said, the ventilation perfusion ratios are very, very important. Then we have the next division, which we refer to as the respiratory portion, also referred to as the pulmonary acini. Pulmonary acini. This is the functional unit of the lung. It's a repeated sort of structure and constitutes the functional unit of the lung. And the pulmonary acini or respiratory portion is where we have the actual exchange of gases, actual exchange of gases. So the respiratory portion, also referred to as pulmonary acini, comprises of the respiratory bronchiole, the alveolar ducts, alveolar sac, and alveoli. Respiratory bronchiole, alveolar duct, alveolar sac, and alveoli. All these gases can be able to be exchanged at any part of this respiratory portion. Then, like I said, we have the ventilatory portion, and the ventilatory portion comprises of the ribs. The curvature of the ribs is very important. The muscles attached to the ribs, the intercostal muscles, are very important. The diaphragm, as the chief inspirator, also very important. And then accessory muscles also play a very important role. These are muscles which are attached to the rib cage, including the abdominal muscles when you are forced expiration, and also the muscles attaching from the upper limb or the vertebra to the ribs or the sternum. So these ones are very important as accessory muscles of respiration. Apart from this, within the internal structure of the lung, we have the elastic tissue. This also plays a very important role in the elastic recoil during passive expiration. During passive expiration. So these are the parts of the respiratory system that we'll be looking at. In terms of the rib cage, that will be addressed in the gross anatomy. In terms of organization, topographic organization of the respiratory of the thorax, that will also be addressed in gross anatomy in terms of the mediastinum and its contents. Divisions of the mediastinum and their contents will be addressed in gross anatomy, including the innervation of the thoracic cavity and thoracic wall and cavity, and also the blood supply to these regions will be addressed in gross anatomy. So let's look at this. We just talked about the pulmonary system, tracheobronchial tree, up to the terminal bronchial. And when you have the diameter reducing to about 0.5, that will be the terminal bronchial. So all these parts here, from the nasal cavity all the way to the terminal bronchial, they are always kept open. So we are going to look at what structures keep this open. They're always kept open, but towards the terminal bronchial, we also have the musculature that can adjust the diameter, causing bronchial constriction or bronchial dilatation. So this will be looked at. And then the respiratory bronchial alveolar duct, alveolar sac, and alveoli constituting the respiratory portion. This is the pulmonary acini. Pulmonary acini from the respiratory bronchial all the way to the alveoli. And we are going to look at these structures in detail. So what is the characteristic or adaptations of the conducting portion of the respiratory system? The conducting portion of the respiratory system. What will be their characteristic histologically? One is it has to be kept open, so it has to be patterned all the time to facilitate movement of air. And this is achieved in the nasal cavity. We have some cartilages and also bones in the nasal cavity. As we move to the trachea, we have cartilages, tracheal cartilages, the C-shaped cartilaginous rings. As we move to the primary bronchi, we have also cartilages, C-shaped. As we move to the lower bronchi, we have cartilaginous plates. And then towards the terminal bronchial, instead we have muscle and connective tissue, keeping this patterned, keeping this open to facilitate passageway of air. So we'll be looking at all this in terms of histology as we look at each of the parts. The other important aspect is air turbulence. We need to mix up the air. So in mixing up this air, what is the achievement of turbulence? In conducting air, the conducting portion does not just conduct the air, it also conditions the air. Air conditioning involves removal of dust particles or filtration, large and small particles. It also involves warming of air so that when the air gets to the lungs, it's at body temperature. It also involves humidification of air, moistening of air. That is conditioning. So apart from conducting the air, it's also conditioning the air. Now, in achieving this, we have structures within the conducting portion, especially the upper part of the conducting portion, that will be able to facilitate this. Air turbulence is an important aspect which causes mixing of air. But in the process of mixing of air, we also have particles being pushed away from the center. If you remember from your physics, in terms of turbulence, centripetal and centrifugal forces acting will push large particles away from the center, caused by the turbulence. So when it's pushed away from the center, it's going to stick to the mucus within the wall of the conducting portion. So we are going to look at where those mucus come from, but it's going to stick to the mucus along the way of the conducting portion. So to achieve this turbulence, the nasal cavity is not a smooth structure. The respiratory passageways are not entirely smooth structures throughout. So we have in the nasal cavity, we have the concha, whose main framework is the turbinate bones. Turbinate bones. So the concha, superior concha, and the inferior concha, middle concha, and inferior concha are folds, lateral folds, projecting from the lateral wall of the nasal cavity. In between this concha, we have some meaty. This meaty also is where the air passes through, but also act as an opening for the paranasal sinuses. Paranasal sinuses, which also play a role in conditioning of air. We have the epiglottis, which guards the opening of the larynx. Now, at the guarding of the opening of the larynx, the epiglottis can create turbulence as the air enters into the larynx. And within the larynx, we have laryngeal folds, true and false laryngeal folds, the true folds, vocal cords, and also the ventricular folds, can cause turbulence of air as it enters into the trachea. And at every bifurcation of the bronchial tree, turbulence of air can be induced in those areas. And this is very important in pushing light particles to the surface for trapping of air. At the opening or at the bifurcation of the trachea into primary and into the primary bronchi, where we call the carina, this is also a very important part that actually turbulence is caused at that bifurcation. I've said at every bifurcation of the tracheobronchial tree, we have turbulence of air. The other important aspect is regulation of airflow. Regulation of airflow. At any given time, your nasal cavity, the two nostrils, do not allow air to flow at the same rate. So they are interchangeable. So that it gives time for the glands and also the secretions within the nasal cavity to be enhanced for recovery. This regulation of air is achieved by the large blood vessels, especially at the opening, at the area of the nasal septum, where we have what we refer to as Kisselbach's plexus. Kisselbach's plexus. We are going to look at Kisselbach's plexus, but Kisselbach's plexus and also the venous sinuses. The venous sinuses within the lamina propria of the nasal cavity proper. At the Kisselbach's plexus and also in the venous sinuses, we have some erectile tissue, some smooth muscle that we refer to as intimal cushions. The intimal cushions within the blood wall can contract, and by virtue of that, it can cause engorgement of the blood vessels within the sinuses, and therefore reduction of the space in which the air flows within the sinuses. Within the nasal cavity and also in the respiratory tract, we have neuroepithelial bodies. Neuroepithelial bodies, these we refer to as the brush cells forming at the basal parts connections with the nerve tissue that initiate this regulation of airflow. So we are going to look at what are these neuroepithelial bodies and what is their role. We have the warming of air, and warming of air, like I've said, is part of the conditioning of air. And warming of air is achieved by, one, an increase in the surface area by the concha. Because of these high scroll-like structures, folds, we have a very big increase in surface area. And immediately in the lamina propria, within the venous sinuses, we have very well vascularized lamina propria. So the heat from the inner parts of the body in these blood vessels will be conducted to the surface, and as the air passes through the surface, it is picking up the heat by convection. So the air is warmed by convection. The heat is transmitted from the blood vessels to the surface by conduction within the wall of the blood vessels and also the structures of the lamina propria. So this particular warming is very, very important, especially in the nasal cavity. The nasal cavity. We also have moistening of air. Moistening of air. If you are to breathe in dry air, our pulmonary acini, which is comprising of a very thin surface, very thin wall, can very easily dry out. So by the time the air gets into the lungs, it should be moist. So the moistening of air is achieved by mucocerous glands in the nasal cavity, in the trachea, in the primary bronchi, and as you move towards the bronchial tree, we have cells that secrete fluid secretions that help in moistening of the air. So apart from the mucocerous glands, we also have the goblet cells, which are unicellular glands. Unicellular glands. Then we have trapping and removal of dust particles. Within our nasal cavity, we have the hair in the vestibule of the nasal cavity, also called vibrissae. This hair helps in trapping very large particles. If you are to have a fly or something gets into the nasal cavity, then that will be prevented by this large hair, very strong hair. So that will be trapping and of dust particles. The vibrissae in the nasal cavity also contains, at the basal parts of the hair, it contains tracheal nerve endings. These tracheal nerve endings can sense any movement of the hair, and if you have something getting into the nasal cavity, it triggers a generation of an impulse, which then causes you to sneeze to be able to remove the light particles that are getting into your nasal cavity. So if you have a fly getting into your nasal cavity, you will tend to sneeze immediately to remove this. And then in the rest of the respiratory passageway in the nasal cavity, in the trachea, we have also mucus lining. The mucus lining produced by the mucocerous glands, this will help in trapping dust particles. Mucus is secreted as mucin by mucus glands. The mucin contains water, then we refer to that as mucus. So the water evaporates. As it evaporates, it moistens the air. And then we have the mucociliary activity, the cilia, which is in the nasal cavity proper, in the larynx, in the trachea, all the way to the terminal bronchi, we have cilia. The cilia, through a mucociliary activity, remove the trapped dust particles. The cilia in the nasal cavity beat towards the pharynx. The cilia beyond the nasal cavity in the larynx, the trachea, the bronchi will beat also towards the pharynx. And once the mucus accumulates in the pharynx, then you either swallow it or you cough. It initiates a swallowing reflex or a cough reflex so that you can remove the excess mucus that are accumulating in the pharynx. Apart from this, any particles, any bacteria, any viruses that might have bypassed this system of trapping of dust particles and particulate matter and bacteria, if they enter into the pulmonary acini, then we have alveolar macrophages. Alveolar macrophages. So these alveolar macrophages will phagocytize any large particles, any bacteria or viruses that will have bypassed the upper respiratory passageway. Apart from this, we also have lymphoid organs. In the lamina propria, we have surveillance system where we have cells of the immune system within the lamina propria that also will help in protection. So protection, we have the lymphoid follicles forming part of the bronchial associated lymphoid tissue, and also what we refer to as the nasal associated lymphoid tissue. Apart from this, we also have the secretions from the respiratory passageway. We'll have secretory immunoglobulin A, which is very important in protection. So bronchial associated lymphoid tissue, nasal associated lymphoid tissue, secretory IgA macrophages are also very important in that protection. Apart from this, the sensory tracheal nerve endings, which I've just talked about, sensory innervations to the pharyngeal regions, sensory innervation to the nasal cavity, especially the hair of the nasal cavity, causing the sneeze or cough reflex. That is a protective mechanism. Then we have resonance of sound. The paranasal sinuses play a very important role in resonance of sound, and also the larynx play an important role in production of sound. Paranasal sinuses are within the bones that surround the nasal cavity, the frontal bone, the ethmoid bone. All those have air within them, and they are lined by similar epithelia like what you have in the nasal cavity, and therefore play a role in conditioning and also resonance of sound. So these are very important adaptations as we look at the various histological structures of the nasal cavity and the respiratory passageway. We need to bear this in mind and look at where they are. Now for the nasal cavity, I've said we divide the nasal cavity into two parts. We have the outer part of the nasal cavity, the nostrils. This part we refer to as the vestibule of the nasal cavity. Vestibule of the nasal cavity is a continuation of the epidermis and the dermis. Epidermis and the dermis. It is lined.
The epidermis is the outermost.
It is lined by stratified squamous epithelia. Stratified squamous epithelia, just like what we have in the skin, in the epidermis. And below the epidermis, we have the dermis. And the dermis consists of dense, irregular connective tissue. Dense, irregular connective tissue. I think we looked at this when we were looking at the skin, dense, irregular connective tissue, or when we are looking at connective tissue in general, we classified connective tissue as dense, irregular connective tissue. Apart from this, we also have the hair and sebaceous glands. Hair and sebaceous glands, just like what we have in the nasal cavity. Sebaceous glands, remember, produce, and sweat glands produce fluid, which also participates in moistening of air. And then, as I've mentioned before, the hair is very important in trapping of dust particles or large particulate matter, and also act as sensory structures that facilitate the sneezing reflex should light particles get to the epithelia. And then as we move towards the inner part, the nasal cavity proper, we have the transition where we have the changes from stratified non-keratinized epithelia to pseudostratified ciliated columnar epithelia with goblet cells. Pseudostratified ciliated columnar epithelia with goblet cells. So the whole of this part is lined by that epithelium. And the nasal cavity proper contains these scroll-like folds from the lateral wall and a septum, nasal septum. The scroll-like folds are referred to as the concha. We have the superior concha, where we also have the olfactory epithelia. We have the middle concha and the inferior concha. In between this concha, we have these spaces, which we refer to as the meatus, nasal meatus. So we have the inferior nasal meatus, then mainly the maxillary paranasal sinus open into. We have the middle meatus, and we have the superior meatus. So these scroll-like folds lined by an epithelia, which is very highly vascularized, increase the surface area, especially for conditioning of air. Within this, we have an epithelia made up of pseudostratified ciliated columnar epithelia with goblet cells. The main cell types within this nasal cavity will be the ciliated cells, which have cilia on the apical surface, and the cilia beat towards the pharynx. So they remove any particles that have been trapped towards the pharynx. Then we have goblet cells, which are goblet-shaped cells within the epithelia, embedded between the ciliated cells. Apart from the goblet cells are unicellular mucus-secreting glands. And then we have the basal cells, and basal cells are stem cells. They are stem cells which differentiate to form the other cell types within the nasal cavity. And apart from this, we have the brush cells. I'd like us to pause briefly, just try to internalize what we have just talked about in terms of the nasal cavity, in terms of structural adaptations for a minute. Fine. So that's what we have as part of the respiratory passageways. Apart from these cells, we also have the other cell types are just described, which we refer to as the brush cells. These are neuroepithelial cells. They're actually columnar cells, and their apical surface have some microvilli, which might act as receptors. And at the basal surface, we actually have attachment to autonomic nervous system. So once these are triggered, this is what can trigger excess production of secretions, should there be so much dust getting into our nasal cavity. Apart from this, the epithelia rests on a very thick basement membrane, and after the basement membrane, we have a lamina propria. And the lamina propria, like we had said before, consists of loose connective tissue. Loose connective tissue with some surveillance immune cells, and also seromucous glands, which we had talked about. These are mucus and serous glands. And these mucus and serous glands are very important in moistening of air and also trapping of dust particles. Within the lamina propria, we also have a very well-developed venous sinuses. These are very large blood pools, and these venous sinuses are important in warming of air, which we talked about, as the air moves by conduction from the venous sinuses to the surface, then by convection, when the air gets in, when the heat is conducted to the surface, when the air gets in, it picks up the warmth by convection. And we had also talked about these venous sinuses are equipped with intimal cushions. And most of these are also in this part here and in the nasal septum, in the what you call the Kisselberg plexus, or the little triangle, which is part of the nasal septum and this anterior part of the nasal cavity. So this way we have very well developed venous sinuses. Now, the area here, the Kisselberg triangle or area, is also an area where you normally have nose bleeding. Because the blood vessels are very large, only covered by a very thin epithelia, then it is very easy for these blood vessels to be damaged. And that's why we have nose bleeding, especially in the anterior part. The venous plexus or Kisselberg's plexus is usually made up of very many blood vessels that form a network within this anterior nasal septum. This network is made up of palatine blood vessels, greater palatine vessels. We are part of the anterior and posterior ethmoidal vessels. We have the labial vessels also forming part of these sinuses. So the sinuses, they are made up of blood vessels from internal carotid and also external carotid. Apart from this is what we call the respiratory part. But in this upper part, in the nasal concha, in the posterior part, we have also the olfactory epithelia. So the nasal cavity, apart from conducting and conditioning of air, also contains the olfactory epithelia. And the olfactory epithelia is made up of a pseudostratified epithelia, with olfactory cells which are bipolar cells, and some sustendacular or supporting cells. So we have olfactory cells, bipolar cells, we have sustendacular cells or supporting cells, and we have basal cells or undifferentiated cells. in this region. Within the olfactory epithelia, in the lamina propria, we have some specialized glands we refer to as olfactory glands, also called Bowman's glands. Bowman's glands. These Bowman's glands in the olfactory secrete some serous fluid, thin serous fluid, in which the odorant, what we are supposed to smell, must dissolve in. So the olfactory bipolar cells, which we'll be looking at later on when I look at the nervous system, will consist of some cilia at the apical surfaces, and the axons from this pass through the cribriform plate of the ethmoid bone, which is part of the roof of the nasal cavity. The cribriform plate of the nasal bone as fila olfactoria. So those axons form what we call fila olfactoria. And immediately they then synapse with structures in the olfactory bulb. In the olfactory bulb within the anterior cranial fossa, within the cranial cavity. Middle cranial fossa, sorry. So let me just repeat for the olfactory. The significant parts is we have olfactory cells, supportive cells, also called sustendacular cells, the basal cells which are stem cells within the epithelia. And remember, the turnover of these cells is very high. So there's usually the stem cells that are to replace the cells. And then in the lamina propria, we have the specialized Bowman's glands. Bowman's glands, also called olfactory glands, that produce the fluid in which the odorant must dissolve in. So that's the difference between what we have in the respiratory part and the olfactory part. In the olfactory part, in the lamina propria, we have Bowman's glands. In the respiratory part, we have mainly mucoserous glands and also the venous sinuses, So that's what we have in the olfactory part. Now, to create patency of the nasal cavity, we have a nasal septum, separating the nasal cavity into two parts, nostrils, the nasal septum. The nasal septum posteriorly is made up of bone, perpendicular plate of ethmoid bone, and then anteriorly is made up of cartilage. So bone posteriorly and cartilaginous anteriorly. And then the lining of the nasal septum is the same as the lining of the concha, mainly what we refer to as respiratory epithelia. So that's what we have as the nasal cavity. And remember, within the meaty, we have openings of the paranasal sinuses, and we say the paranasal sinuses are very important in conditioning of air and also some resonance of sound. This is the ethmoid bone, sphenoid bone, sorry, and this is air space, the sphenoid air sinuses. Sphenoid air sinuses. We have sphenoid air sinuses, we have the maxillary air sinuses, which are very big. Maxillary is very, very large. Then we have the pharynx. This is the pharynx here. And the pharynx, as I've said, is divided into three parts. We have the nasopharynx, we have the oropharynx, and then the laryngopharynx. Within the nasopharynx, we have the opening of the eustachian tube, also referred to as the pharyngotympanic tube. Pharyngotympanic tube. And this pharyngotympanic tube is guarded by some lymphoid tissue. Lymph nodules we refer to as the lymph nodules, adenoids. These lymph nodules are very important. The tubal tonsils, we call them tonsils, are guarding the opening of the Eustachian tube. And then within this area of the uvula, we also have the tonsils, this lymphoid tissue within the uvula. In the nasopharynx is lined by something similar to respiratory part, which is pseudostratified ciliated columnar epithelium. In the oropharynx, on the other hand, because the food is passing through this area, you need to have something that is strong, and therefore we have in the oropharynx lining by continuation of the oral mucosa, which is stratified squamous epithelia. Stratified squamous epithelia. And then we have immediately below that stratified squamous epithelia, we have some lamina propria, and we have these pharyngeal constrictors, the superior, middle, and inferior pharyngeal constrictors. These pharyngeal constrictors are to help facilitate in swallowing process. And then the laryngopharynx is lined by respiratory epithelia. The upper part of the epiglottis will be lined by the stratified squamous epithelia. The lower part of the epiglottis will be lined by respiratory epithelia, which we had just described. So that is about the nasal cavity, and the nasal cavity is very, very important in conducting and conditioning of air. Within the pharynx, we also have pharyngeal tonsils. Pharyngeal tonsils, what we refer to as Weldear's ring. Weldear's ring. This is an accumulation of lymph nodules, lymphoid tissue that constitutes the Weldear's ring. This comprises of the tubal tonsils. We have the lingual tonsils at the base of the tongue. We have the palatine tonsil at the palatal fossa. And we also have the pharyngeal tonsils, where at the uvula, we have part of the pharyngeal tonsils. So these are very important in guarding this region, because this is the opening to the ear, the opening to the nasal cavity, to the respiratory system, and also the opening to the digestive system. And therefore, it's very, very important to have a lot of lymphoid tissue to be able to guard and initiate, usually activation, trapping antigens, and initiation of immune system. Apart from this, in the lamina propria, like I've said, we normally have some surveillance lymphoid tissue. So this is what I've talked about for the blood supply and also nerve supply that will be covered in gross anatomy. This is part of the nasal cavity. Here we have some concha. These are the scroll-like openings within the nasal cavity. What appears here as red is bone. This is bone, which is continuation to the maxillary bone and also continuation to the ethmoid bone and so on. So this one is here bone, and lining this bone is a mucosa. We refer to this as a mucosa because it's opening to the outside. It's a nasal mucosa. And the nasal mucosa will be comprising of, here we note two parts. This part is the respiratory part. This part is the respiratory part. This part here is the olfactory part. In the olfactory part, we have a very thick pseudostratified with columnar cells. We have the olfactory cells. We do not have, in the olfactory part, we do not have goblet cells. In the lamina propria, we have this. This is blue staining. These ones here are the Bowman's glands. Bowman's glands. In the nasal respiratory part, we do not have the Bowman's glands. Instead, we have the venous sinuses. Venous sinuses and mucocerous glands. So that's the difference between the olfactory and the respiratory parts. Olfactory and respiratory parts. So this is just an illustration. We've talked about them. This is the lining of the nasal cavity at the magnification. Remember, there is this bone. Then all these spaces here are actually filled with blood. These are the venous sinuses, and within the wall of the venous sinuses, we have the intimal cushions. Intimal cushions which regulate the flow of blood. Then we have the epithelia with a lot of goblet cells. Goblet cells, pseudostratified ciliated columna with goblet cells, columnar ciliated cells, basal cells, and brush cells. Since we have described the nasal cavity, it also is lined by an epithelia that we refer to as the respiratory part of the nasal cavity. We can look at what is the general description of our respiratory epithelia. We are going to refer to this word respiratory epithelia in the trachea, the primary bronchi, the lobar bronchi, the tertiary bronchi. We'll use the word respiratory epithelia because it's common to all these parts. So what constitutes respiratory epithelia? It is usually pseudostratified ciliated columnar epithelia with goblet cells. We have the ciliated columnar cells, very rich in mitochondria. Remember, the mitochondria is to provide mitochondria is to provide the necessary force for the cilia to be able to move. We have rough endoplasmic reticulum. We have smooth endoplasmic reticulum, lysosomes, and Golgi apparatus. These are the predominant organelles that we have within the ciliated columnar epithelia. We also have the basal cells, and basal cells, I've said, are stem cells and differentiated cells. They are polygonal cells with very large nucleus. Very large nucleus. And these polygonal cells will have usually undifferentiated organelles. Undifferentiated organelles. Because they're going to differentiate to form goblet cells and also going to differentiate to form ciliated cells and so on. So these are stem cells. Remember, the epithelia in the nasal cavity is replaced by every 40 days. It's completely replaced, some of the epithelia, or even shorter. Intercalated cells, which we are like in the process of differentiation. In the process of differentiation, these cells of the intercalated cells would also constitute part of the brush cells. The brush cells. So the brush cells have microvilli on their luminal surface, microvilli on the luminal surface, and they make conduct synapses with autonomic nervous system, and therefore very important in monitoring the structures within the epithelial lining, so that if there is any irritant material within the epithelial lining, then we can have excess production secretions to be able to dilute that irritant material. We have goblet cells, and goblet cells are goblet shaped. They are goblet shaped. They have an expanded apical part, which is full of mucus. We have a narrow basal part where the nucleus and the organelles are located. We have bright cells. I've talked about them as cholinergic nerve endings. And we have neuroendocrine cells. Neuroendocrine cells usually secrete hormone-like structures, mainly peptides, that can have either paracrine activity, would influence the cells immediately next to it, or autocrine activity. So the secretions can influence the cell which is producing it, because they have receptors within the cell. As we go down the respiratory tract, we'll be looking at Clara cells. Let's leave that for now, but deal with these upper parts here. Now, olfactory mucosa is up to 500 millimeters square. I've talked about them in terms of its location in the superior concha. We have olfactory cells that are bipolar, the terminal dendrites, and we have what olfactory vesicle is an expanded part of the bipolar cell. And from there, we have olfactory cilia, which run parallel to the surface. The olfactory cilia originating from the olfactory vesicle actually run parallel to the surface. And it is this cilia that the substances that have to be dissolved attach. They attach. We also have sustendacular cells or supportive cells, and these are columnar cells, supranuclear Golgi apparatus because they secrete some substances, and the terminal web. This terminal web consists of microfilaments, which are very important in supporting these olfactory cells. And the microvilli on their surface will also have some actin at their core. So they have actin at the core of microvilli that attach to this terminal web. So the terminal web is to increase the strength of the cells at the apical part and therefore protect the olfactory cell. And then we have basal cells, which are undifferentiated cells. And then in the lamina propria, I've talked about Bowman's glands. Bowman's glands secreting some serous and mucus fluid that are very important in dissolving the odorant, something that you are going to smell. Mm-hmm. For the larynx, this is the laryngeal muscles, the laryngeal constrictors and dilators. So we have adductors and abductors of the larynx. This will be covered under histology. What I wanted to show here is this vocal cords, which is the true vocal cord. We have two folds within the larynx. We have the true vocal cords and we have the ventricular folds. The true vocal cord, remember, is the one which vibrates, and as it vibrates, it produces sound. Because of that, a lot of forces are being acted on it. So the epithelial lining of the true vocal cords will be stratified, squamous, non-keratinized epithelium. Stratified, squamous, non-keratinized epithelium. The rima glottitis is the apical opening between the true vocal cords, opening between the larynx into the trachea. So by adjusting the diameter of the rhimaglottitis and by forcing the air through, you are able to generate different sounds. And this is possible because we also have the tensing of the vocal cords. We have the main framework of the vocal cord is a very dense connective tissue membrane that we call the vocal membrane. And lining this vocal membrane, we now have the stratified squamous non-keratinized epithelia. The muscles of the larynx and the cartilages of the larynx the cartilages are supposed to provide the patency to make the larynx open. The muscles also, apart from making it open by abducting, also adjust the diameter of the rhimaglottitis and therefore tensing the true vocal cords and causing vibration. Just posterior to the larynx, we have the esophagus. We are going to look at this in terms of structure. You will note the esophagus here is very close to the larynx and also very close to the trachea. But for the gross parts, you'll be dealing with those. So the laryngeal cartilages give patency, the muscles adjust the diameter, and then the mucosa is not respiratory mucosa only in the true vocal cords. The rest of the larynx is covered by a respiratory mucosa. So you know what a respiratory mucosa is. The vestibule of the larynx
except
the
vocal
upper
cords.
part of the larynx. Please, can you mute your microphone?
Yes.
So the innervations here will be, gross anatomy will be talking to you about them, but you're talking about the laryngeal nerves, laryngeal nerve branches of the vagus nerve. For the trachea, the rhimaglottitis opens into the trachea. The trachea, just like what we have in the nasal cavity, is lined by respiratory epithelia. So the mucosa of the trachea is made up of an epithelia that is respiratory. Just to remind you, it's made up of pseudostratified, ciliated, columnar epithelia with goblet cells. So we have ciliated cells, goblet cells, brush cells, basal cells. We have a very thick basement membrane, in which the epithelia rests on. And then we have the lamina propria. What appears blue here is connective tissue. The lamina propria that's made up of loose connective tissue, having some lymphoid tissue, and also the openings of the ducts for the openings of the glands. In the submucosa, so this is the mucosa. In the submucosa, we have dense connective tissue and some mucocerous glands. These are the mucocerous glands made up of serous acini and also mucus acini. Serous and mucus acini, because it's a mixed gland, we refer to this as mucocerous glands. And these are the ones that produce fluid that is going to moisten the air, and also trap dust particles. The next layer is a layer we refer to as fibromusculocartilaginous layer. Fibromusculocartilaginous layer. It's made up of C-shaped cartilaginous rings, incomplete C-shaped cartilaginous rings made up of hyaline cartilage. The hyaline cartilage is covered by perichondrium. Between the hyaline cartilage, because they are C-shaped, as you go, there are several cartilaginous rings. Between the cartilaginous rings, we also have some fibrous connective tissue sheet that forms what we call lamina fibrosa. In the open part of the C-shaped cartilaginous ring, we have the trachealis muscle, which is a smooth muscle. Trachealis muscle, which is smooth muscle. And then the outermost layer is referred to as the adventitia, tunica adventitia, made up of dense connective tissue, also having the large blood vessels and the nerves that supply the trachea. So the trachea is made up of the mucosa, submucosa, fibromusculocartilaginous layer, and adventitia. So that's what we have for the trachea. Now, behind the C-shaped, the open part of the C-shaped cartilaginous rings point posteriorly. And lying very close to that is the esophagus. Points posteriorly, and lying very close to that is the esophagus. Remember, esophagus is posterior to the larynx. And the idea that the esophagus lies posterior to the open part of the C-shaped cartilaginous rings is to facilitate swallowing. Because that area, there is no cartilage impinging or protruding towards the esophagus. The trachealis muscle, innervated by the autonomic nervous system, can adjust the diameter of the trachea. And this also extends to the primary bronchi. After that, we go to the secondary bronchi, where at the carina, at the position of the carina, we actually have a complete ring of the C-shaped cartilaginous ring at the carina. So it forms something complete. And then at the primary bronchi is the same as the trachea in terms of structure. The secondary structure, but as we move to the lobar bronchi, we now have not cartilaginous rings, but we have cartilaginous plates. Cartilaginous plates replace the cartilaginous rings. The epithelia is still pseudostratified columnar epithelia, ciliated columnar epithelia. The glands in the submucosa are still the same. Instead of the cartilaginous rings, we have cartilaginous plates. We have an adventitia that merge with the rest of the tissues around. So this is what we have also in the lobar bronchi. Then as we extend within the respiratory system, the secondary bronchi, these are secondary bronchi. We have cartilaginous plates which are not hyaline, but are elastic. Some isolated cartilaginous plates that are elastic in nature. The mucosa is still pseudostratified ciliated. The number of goblet cells is reduced. Instead, we have what we refer to as a bronchiolar epithelia. And that epithelia comprises of ciliated cells, still columnar cells, the neuroendocrine cells, and some Clara cells. Clara cells. Now, Clara cells are very rich mitochondria because they are very important in movement of material inside the cell, intracellular transport. They secrete some protein, surfactant protein, and therefore are very important raffinoplasmic reticulum for secreting the protein, surfactant protein. The smooth endoplasmic reticulum in secreting lipids because the surfactant consists of lipids also, and also secretion of glycosaminoglycans and glycerol. Glycosaminoglycans and glycerol and surfactant protein. Apart from this, the main function of the Clara cells is also main function that's important is also in detoxification. Detoxification. So they play a role in detoxification. And this extends from the secondary bronchi all the way to the terminal bronchi. But as we move to the terminal bronchi, we do not have any more goblet cells. We do not have any more goblet cells. We have a bronchiolar epithelia. The columnar cells have now reduced and become some simple cuboidal ciliated cells. Simple cuboidal ciliated cells. And these simple cuboidal ciliated cells are able to, again, trap particles and push them out towards the larynx. Simple cuboidal ciliated cells will be resting on a very thin basement membrane, and immediately below this basement membrane, we have some smooth muscle and also some elastic cartilage. I mean, elastic fibers, smooth muscle, elastic fibers, and some collagen fibers. But predominant among these cuboidal cells are also the Clara cells. The Clara cells. We have Clara cells, and the main functions of Clara cells, like I've said, is secretion of the glycosaminoglycans, secretion of surfactant proteins, specifically surfactant protein A and D. A and D. A and D are very important in immunomodulation. Immunomodulation, that's protection. And then detoxification. We have talked about detoxification. And Clara cells can also act as basal cells. They are undifferentiated cells that can differentiate to form other cells within the bronchioles. So in the terminal bronchiole, that's what we have. And that is the end of the respiratory bronchiole. So this is what we have. There's no goblet cells here. We have smooth muscle and also some collagen fibers here. And around it, the adventitial part is continuous with the connective tissue of the lung. So we have a lamina propria, mainly having connective tissue, a lot of elastic fibers to facilitate some flexibility, and then smooth muscle. Supplying these smooth muscles, we have mainly sympathetic nerve endings, sympathetics, both parasympathetic and sympathetic nervous system to regulate the diameter. That's what we call bronchial constriction, and that is very important. And then from here, we now move to, as the diameter becomes smaller, we now move to what we call the respiratory bronchiole. Respiratory bronchiole. So from the terminal bronchiole, we move to respiratory bronchiole. These are respiratory bronchiole. Now, the respiratory bronchiole is a discontinuous tube. Discontinuous tube lined by simple cuboidal epithelia. Simple cuboidal epithelia. Also having some Clara cells. The simple cuboidal epithelia rests on a basement membrane, and in the lamina propria, we have some smooth muscle and a lot of elastic fibers. Now, remember that in the respiratory bronchiole and also in the terminal bronchiole, we do not have any cartilage. Instead, the smooth muscle and the collagen fibers are playing a role in providing patency of the airways. Patency of the airways. Now, the respiratory bronchiole, we said at the respiratory bronchiole, this way we have now what you call the pulmonary acini. So this is the terminal bronchiole, which divides severally into a respiratory bronchiole. This is a respiratory bronchiole. In this respiratory bronchiole, I've said, is a discontinuous tube lined by simple cuboidal epithelia. At these areas, we have simple cuboidal epithelia, some smooth muscle and some elastic fibers and collagen fibers in the lamina propria. And the tube is studded by alveoli. So we have alveoli opening into the respiratory bronchiole. Because of that, we now refer to this part as the beginning of the respiratory division of the respiratory system, because you can actually have gas exchange taking place within this respiratory bronchiole. So it is started with some alveoli opening. From the respiratory bronchiole, it divides severally, sometimes up to 6 times, into what we call an alveolar duct. This is something similar to an alveolar duct. An alveolar duct does not have a wall of its own. It actually comprises of the openings of the alveoli. The openings of the alveoli, mainly made up at that opening by a lot of elastic tissue. Elastic tissue, a bit of collagen fibers, but predominantly elastic tissue. So that's what we call an alveolar duct. The whole of this part here is an alveolar duct. And the terminal part of that alveolar duct is what we refer to as an alveolar sac. Now, an alveolar sac is where several alveolar ducts open. This part here could be having up to 4 or 5 alveoli opening. So this is what you call an alveolar sac, lined by not having a wall of its own, but merely the opening of several alveoli into a common lumen. And then finally, in the respiratory part, we have the alveoli. This is an alveolus. This is an alveolus, and this is the smallest part where gas exchange takes place terminally. It's made up of a double wall of two alveoli. Between the alveoli, we have interalveolar pores. Between this one and this one, we might have a hole connecting, but between the alveoli, we have interalveolar pores. P-O-R-E-S. These are the opening connecting two alveoli, and the important part of that is to equalize pressure. So what are the adaptations of this respiratory division? What are the adaptations of the respiratory division? To be able to have gas exchange, what are the adaptations? The cells have a very, very thin wall, very thin. They are highly attenuated cells, very highly attenuated. Squamous cells, they are simple squamous cell that is stretched and very, very thin so that gas can diffuse through from the blood to the lumen. We also have a reduction in blood-air barrier, the space between the blood within the capillaries and the air within the lumen of the alveoli. The blood gas barrier or blood-air barrier is made up of the following structures. From the alveoli, we have the hypophyse, which is a fluid lining the inner part of the alveoli, type 1 pneumocyte, alveolar type 1 pneumocyte. We have the alveolar type 1 pneumocyte. So we have the fluid film, type 1 pneumocyte. We have the basement membrane of type 1 pneumocyte. We have the basement membrane of the endothelial cell, which is also very thin. The endothelial cell is the cell that forms the blood vessel. And then we have the endothelial cell, and the inner part of the endothelial cell is lined by an endocalyx. And then you go to the lumen of the blood vessel. So let me go in reverse. From the lumen of the blood vessel, you have an endocalyx, endothelial cell, basement membrane of the endothelial cell, basement membrane of the type 1 pneumocyte, then type 1 pneumocyte, and then the hypophyse, some fluid film lining the inner part of the alveoli. In the extreme case, we have actually the basement membrane of the endothelial cell and the basement membrane of the type 1 pneumocyte are fused together. So we have a common basement membrane to reduce the distance between the lumen of the blood vessel and the alveoli. We also have an increase in surface area. We have millions and millions of alveoli. Millions and millions of alveoli. If you take all the alveoli and stretch them out, they form over 143 to up to 150 square meters of surface area. So you can see a very, very large surface area. So that promotes gas exchange. We have an increased vascularization. Almost every type 1 pneumocyte is next to the blood vessel because we have a very rich anastomosis of the capillaries. These are the continuous type of capillaries. Continuous type of capillaries. So all these factors are very important in gas exchange. And that's why we refer to this part from the respiratory bronchial to the alveoli or the pulmonary acini as the functional unit of the lung, because the main purpose of the lung is for oxygenation of blood. Apart from this, we also have protection, and protection is achieved by the presence of these black cells here. These are actually alveolar macrophages. Alveolar macrophages. The alveolar macrophages are actually on the surface of the alveoli. They are moving around the surface of the alveoli. They are also in the interalveolar space. Between the alveoli, we have alveolar macrophages. So this can phagocytize any large particles. Apart from that, the surfactant secretions, which is protein, surfactant protein A and B, are very important in immunomodulation. They promote the immune system and therefore provide protection. Just like what we had seen, protection in the upper parts, but here we do not have that secretory product, secretory IgA. This is an illustration. You'll be looking at this slide in your histology. This is an illustration of the type of vascularization. These are actually, we have injected some latex into the blood vessels, and these blood vessels are between the alveoli. So you can see it is very well vascularized, very rich in blood vessels. To increase perfusion, if we have a cast, corrosion cast injected into, we have injected a corrosion cast into the respiratory passageway from the large passageway. This is for blood, and this is the trachea. So these are the carina. And remember, at every bifurcation, we also have blood dividing. So that terminally, we have the pulmonary acini. So that's why we call this a bronchial tree. Tracheobronchial tree also mimics the capillary network within the lungs. This is just an illustration of the capillaries that completely surround the alveoli, therefore increase perfusion. This part is actually the pulmonary acini. Pulmonary acini. So we have several acini supplied by blood, and therefore this is repeated and constitutes the functional unit of the lung. We looked at that before, and the alveolar ducts. Let's now look at the alveoli. The alveoli, or alveolus, if it is one, I've said it's 143 square meters. If you take all these alveoli and stretch them out, they form 143 square meters of surface area. And we have up to 300 in the adults, up to 300 million alveoli. So there are so many alveoli, which is important. And then we have, this is how thick it is. the thickness, 200 micrometers, very, very thin, so that you can have blood vessels, dust entering. This is the wall of the alveoli. Actually, what you're seeing here is two alveolars, the lining of this alveoli and this one, and we have an interalveolar space. We are going to see what the interalveolar space is. And the alveoli comprise of two primary cell types. We have the very thin cells, which are the type 1 pneumocyte, alveolar pneumocytes. We have the cuboidal cells, which are the type 2 pneumocytes. So we have the type 1 pneumocyte, mainly for gas exchange, and we have the cuboidal cells, which are the type 2 pneumocytes for production of the surfactant. Comprising of surfactant protein A, B, C, and D. Remember, surfactant protein A and D is also produced by the Clara cells, and that is very important in protection, immune modulation. Surfactant protein B and C is very important in maintaining the patterns opening up of this, lowering the surface tension, and therefore opening up, keeping the alveoli open. It's like a soap bubble. All this is produced from glycerol. And if you remember, Clara cells also produce glycerol, which is very important in now synthesis of the surfactant, which is a phospholipid. So the alveolar type 1 pneumocytes are very, very thin. They occupy 97% of surface area. 97% of surface area. Squamous, very flat. Not just squamous, but highly attenuated. Very, very flat. And then the very thin ends of these cells have some very tiny vesicles, what you call micropinocytic vesicles. And sometimes these micropinocytic vesicles join up to form channels, what you call micropinocytic channels. The micropinocytic vesicles is evidence of the movement of passage of the surfactant into blood. So the surfactant is being removed into blood. The wasted surfactant is being removed into blood. They have organelles like any other, and they have a round nucleus, rather flattened nucleus, and they form the blood-air barrier. And I've talked about this blood-air barrier before, where from the blood vessel we have a thin film lining the blood vessel we call an endocalyx. We have the endothelium lining the blood vessel, a common basement membrane of the endothelium and the type 1 pneumocyte, and then the inner part of the type 1 pneumocyte contains hypophyse, which is mainly a thin film of surfactant fluid. So that's what we have as type 1 pneumocyte, specifically gas exchange. The type 2 pneumocyte has several functions. One is it produces surfactant, which is a phospholipid comprising of surfactant proteins and lipids. Remember, if you want to form an air bubble, you don't form an air bubble from water. You can usually use soap So you use soap, put it in water, blow, and you produce air bubbles. Why is soap producing air bubbles? Soap is made up of lipids, it's made up from lipids. That's why you're able to produce bubbles. Similarly, the surfactant that is produced by type 1,2 pneumocyte is very important in forming a bubble-like structure lining the inner part of the alveoli and therefore prevents alveoli from collapse. Sometimes, if there is no alveoli, when the child is born, the lung does not open up. It does not open up. Then we have a respiratory distress syndrome. Respiratory distress syndrome. So the pneumocyte type 2 consists of 50 to 10%, contribute only 3% of surface area. They are round or cuboidal cells, and the surface pointing towards the alveoli have microvilli. But of importance, they have these vesicular lamellar bodies. The lamellar bodies, they seem to be in layers. The lamellar bodies contain phospholipids, which is actually the content for the surfactant. This phospholipid is released by the process of exocytosis into the lumen. So ask yourself, knowing the cell, you know how the cell looks like. Knowing the cell, knowing the product of the cell, it is proteins and lipids. These proteins are not used by the cell, but they are released to the outside the cell where they are used. What organelles, what type of organelles do you think the type 2 pneumocyte should have as predominant organelles? They are proteins for export. So you must have rough endoplasmic reticulum that contains ribosomes, rough endoplasmic reticulum. But these are lamellar bodies. They have some membrane bound initially. And therefore, those membrane bound are not going to be released with their membranes. So they will have a little bit of smooth endoplasmic reticulum to form these membranes. Proteins must be packaged with the lipid production. Lipids are formed by smooth endoplasmic reticulum. So the lipids are formed by smooth endoplasmic reticulum. The proteins are produced by rough endoplasmic reticulum. And all these are packaged together by the Golgi apparatus. So we must have very well developed Golgi apparatus. Where do you think the Golgi apparatus should be? Should it be close to the lumen, above the nucleus, close to the lumen, or below the nucleus, towards the base? Obviously, as you package, you want to transport the product to the apical part, which is close to the lumen, and therefore, the Golgi apparatus must be supranuclear. It's above the nucleus. You are moving substances within the cell. So when you move substances within the cell, you are removing the proteins, you are removing the raw product from the protein, amino acids, you are removing them as a secretory product, So when you move them as secretory product to the Golgi apparatus, and subsequently away after combining with the lipids in the Golgi apparatus, you need energy. So microtubules are very important in moving this. Microtubules. These microtubules need energy. The energy is provided by mitochondria. So those should be the predominant organelles within the type 2 pneumocyte. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, mitochondria, and microtubules. Apart from that, there'll be other organelles, but the predominant ones. The cell is very active, so mitotic activity. Apart from production of the surfactant, the type 2 pneumocyte are also very, very important as stem cells. Just like Clara cells were acting as stem cells, the type 2 pneumocytes also act as stem cells and differentiate to produce type 1 pneumocyte. So they produce type 1 pneumocyte. So the predominant functions, production of surfactant, type 1 pneumocyte. And then of course, there'll be the functions of surfactant. There'll be functions of surfactant. So the proteins are very important. The surfactant proteins are very important in reorganization of the surfactant into what we call tubular myelin. Tubular myelin, and tubular myelin is very important in facilitating that transport of gases across. Now, another important function is formation of type 1 pneumocyte, which I said act as stem cells, but we are going to look at that. Now, when do we say, at what stage do we say this fetus should be ready? At what stage? We look, if we take amniotic fluid, look at the lecithin sphingomyelin ratio. We should have a 2-to-1 ratio in the amniotic fluid of lecithin sphingomyelin. When we have 2-to-1 ratio, then we can say the surfactant is adequately produced for that fetus to survive. So we can do amniocentesis and be able to determine this. Within the lumen, we have alveolar macrophages and also in the alveolar space. And these are mainly monocytes, remember. They can also undergo mitosis to produce themselves, and they are mainly for phagocytosis. If macrophages are for phagocytosis, what should be their predominant organelles? They should have lysosomes. Lysosomes are very important in degrading what is phagocytized. Lysosomes contain enzymes. Enzymes are proteins. So it should be able to have rough endoplasmic reticulum and also some free ribosomes. Transport, intracellular transport. We should be able to have some microtubules and mitochondria. Remember, the lysosome is produced from the Golgi apparatus. It's produced from the Golgi apparatus. So Golgi apparatus is very important. So alveolar macrophages should have very well developed Golgi apparatus. So function is phagocytosis of bacteria, viruses, and fungi, which have been able to get into the alveoli. But also some blood, which has been able to escape. Red blood cells that have been able to escape from the capillaries can also be phagocytized. Clinically, you can have particles being phagocytized